Good morning. Are you able to hear? Good morning. Are you able to see the screen?
Yes, ma'am. Yes, ma'am.
So today we are going to um discuss the topic of forensic entomology, like application of insects to the legal issues, medico-criminal forensic entomology, and arthropods, how they are associated with the crimes, usually violent crimes such as murder, suicide, and rape. So physical abuse and controlled trafficking. So there will be an death investigations regarding these things. So for this, we are establishing the timelines like postmortem interval or PMI, movement of corpse, manner and the cause of death, and association of suspects with the death scene, detection of toxins or drugs, insect larvae. Actually, this forensic entomology we first introduced by the Bergeret in the Western world.
Yes, ma'am.
In the Western world, like forensic entomology was not born until 1855 when Dr. Bergeret used entomological knowledge to help the police in Paris to solve the murder of a child. So, in various specializations, we across this forensic science as forensic toxicology, forensic ballistics, forensic physiology, cyber and digital forensics, document fingerprinting, forensic biology. So, in what way this entomology will be useful for the study of the history of body after the death? So affects estimates of time since death ID of the body and ability to determine the cause and manner of the death. And it also examines the decomposition of the body after the death from individual, cultural, and environmental factors. So what is an forensic taphonomy is very broad and it includes archaeology, biology, earth sciences are all useful. And types of evidences what we have to be collected in this taphonomy is taphonomic evidence. We collect the age, sex, body weight, properties of bones, cause of death, even burial practices, alterations due to unusual circumstances like war, environmental factors like temperature, moisture content, pH levels, properties in the soil. And three main areas of postmortem drug testing, workplace, drug testing, and investigation of contraband materials. And even the entomology is regarding this particular toxicology where we use blood, urine samples, bile, liver, hair as specimens for the testing. So, what are the flies which we play an important role in this particular forensic studies? The Jarder beetles, moth flies, cuttle flies, musset thieves, blowflies, flesh flies, meal moths, fruit flies, etc. So, the Postmortem interval identifying the types of species of insects, rate of development, tampering with results, season, temperatures, and exposure, etc. So, whenever a body is for the investigation, the first responders which are come as an to know the dead of that person or any body, whatever they found, the blowflies, and second responders will be flesh flies. And we'll collect this particular insect specimens as an hand net, forceps, and digging tool, thermometer, vials, jar, plastic bags.
మీ కొడుకుతో
And
విన్నాడు కదా
supporting data needed is of previous weather for area on site weather data,
ఏం
like,
చెప్పాలత్తమ్మా ఏదో
and we need and the particular flies to be whatever they have been collected will be kept in an ambient temperature, maggot mass temperature plays an important role, and the ground surface temperature, and also we see the temperature between the body and surface,
ఆ ప్లేట్ ఇది
of the soil of the identified body, weather data from 1 to 2 weeks period, including rainfall, and maximum and minimum temperatures are taken into consideration while doing the investigations. So, whenever there is an investigation, they
అదే
will
ఒక్క
collect the
మిరపకాయ ఇట్లా
method and
కోసేయండి
living samples,
ఒక్క మిరపకాయ
collect the soil
సన్న
samples, collect the 2 to 3 inches of topsoil.
ఇంకేసి ఇయ్యో
One of you mute yourself. Mute yourself. Collect the soil sample from the under and the adjacent to up and 2 3 feet from the body. All samples placed in a cardboard container for the shipment, and the where this cadaver is very rich but the short-lived resource we will find, and tremendous competition among the organisms we find during that particular investigation, especially in the early stages of decomposition. And we also see the lot of insect colonization of a corpse occurs in the series of stages, and different groups are adapted to different decompositional stages of a corpse. And the stages of decomposition, when we take into consideration, fresh stage will be of days 1 to 2, where the commence of that occurs when bloating is first evident, what we see. And if it is at bloated stage, we see from day 2 to 6. Here the body starts putrefaction and the gases produced by anaerobic bacteria. Decay stage will be 5 to 11. Abdominal wall breaks, allowing the gases to escape. Carcass deflates. And post decay stage will consider from 10 to 25 days. In dry habitats, remains are skin, cartilage, and bones. In wet habitats, wet viscous material in the soil under the remains. And major group of insects associated with the cadavers: blowflies, flesh flies, houseflies, cheese skippers, beetles like carrion beetles, germstids, scarab beetles. All these we find. And when we see about the blowflies, they are blue, green, metallic colored, and they are very much um, uh, related to the housefly body as it is, but they will be in metallic, non-metallic brown form, like the color will be to identify. And green beetle, like some other species, will also be there, where this particular blowflies also will be seen um, near them. And we also see the house flies, flesh flies. The flesh flies can be identified by the yellow spots on the body, and the head region also we find the yellow markings on the head region. Cheese skipper is totally a different fly with the transparent wings, and it also very particularly uh, around the dead bodies we see of 10 years old like that, if it is there lying there only. And we also see the carry-on beetles. Carry-on beetles will be having a red spottings on the body, and these beetles were be arriving to the corpse, and they have a powerful jaws, and they also have a chewing mouth parts, and they also leave a semi-liquid material on the body. And another beetle what we see is an sylphids. This sylphids will be of having an the body with the blackish color, and a little bit yellowish colored wing like structures, and antenna also with the yellow colored hairs. And we also see the beetles belong to the Histiridae family, and also to the Nitidudelidae family also. These are like the like same like an row beetles, and these also like they are having an many of the species lay their eggs in the corpse usually. And we also see the hide beetles, ham beetles. Ham beetles are very common in the decomposition carcass, and they are also metallic sheen colored yellowish body to the anterior side and black body to the posterior side. And they also have the completely dry structure. And this blowfly life cycle, when we see the blowfly cycle, have a six parts: egg, three larval stages, pupa, and the adult. When we come across hours of what the life cycle will be means, when the egg is laid, after 23 hours, we see the pupa stage. After the 27 hours, and on the whole, like around 130 hours. 130 hours means like 5 to 6 days, we see the pupal enclosure. Pupa will be completed, and it will be included in larva will be completed and enclosed into the pupal shell. So if we found pupal shells on the body, we conclude that the body is lying there, that dead body is lying there before 1 week onwards. So that is an indication where we will estimate the depth of the body, how many days since it is lying in that particular place. So when we see around this blowfly lays 250 eggs, and within the after laying of 24 hours, it starts emerging out the from the egg. We see the maggots, and on that, if the temperature is maintained around, there is very convenient temperature is present, then the maggots will grow feeding on that particular dead body. And they have an property of feeding voraciously. They will feed voraciously the dead body, and we see the succession of decay body from day one to day two dry condition, and after the third, fourth, fifth days, they have a different things finding on the body. So the fresh stage blowfly is detected with the on the nostrils of the dead body, mouth, like that, bloated when, and they have an ovipositing swell putrefied, and decay stage feeding, drying out maggots, leaving have left, and we will find postmortem interval. They have an elapsed time from death to the discovery of the cadaver. We see the flies, especially belonging to the determining the postmortem interval. So this fly development, when we see, it requires certain amount of time to complete the temperature dependent, and each species has a certain developmental rate at any given temperature. So two different ways to use the collected data is isomegalon diagram and cultural rearing are the best situations to detect with the best solution. And we also see the collect sample from the crime scene and determine the what is the value from the total required species to determine the unknown amount of accumulated of oviposited flies, eggs, or maggots, or any other stage. And we see the count back the days prior to collection. And we also see the determining the postmortem interval with the step one of collect samples of the insects present in that particular condition. And also we also say that determine the temperature of the history at the crime scene, estimate the time of egg laying, and also we say that what are the other insect evidence we can come to the conclusion other than the beetles. So when we say the stages of decomposition, we come to the conclusion like fresh body is of 1 to 3 days where eggs will be collected. If at all the body is bloated, swelling, bloat, leaking fluids, we'll find. The odor also we see 2 to 7 days we will be concluding larval stages. Decay with the strong odor putrefaction, that is 1 to 3 weeks, all stages. And dry skin and bones we will find in the 1 to 3 months, empty pupae and beetles. So when we see the when we see the development, what are the development, it requires structured into stages, means each stage requires certain amount of time and time to complete a time. Time to complete stage is temperature dependent, and at warmer temperatures, the rate of development will be very fast, and at cool temperature, it slows down. So the each species has a certain developmental rate at any given temperature. And in the like that, when it is going on happening between the stages, even when we see the dry stage thing, in the dry stage, skin and bones and the insects with the ability to digest keratin, clothes, moths, and dermestid beetles will find. And we also see the beetles having an laying of several dozen to thousand eggs also. And this laid eggs will be in clumps. And this particular larvae will have an predatory, they can be predatory. And this larval period species can also be live for several months to years. And this beetle larvae, which are developmental stages we are calling, will be having and several molds in their life cycles. And this, the instar, whatever the instars we are saying, the species will be having so many stages. They'll have a dramatic changes occurring according to the temperature in the surrounding environment. And when we see the beetle larvae pupate, from this pupa emerges a fully formed, sexually mature adult beetles, also sometimes imagos stage like that. So when we are saying about this dry conditions, we even see the beetles having an the chewing mouth parts which can manage the tougher foods to be taken into the for exploitation. And several beetle type make their living out of the copse or nearby to the copse also. And some of these species lay their eggs in the copse and use their powerful jaws to make the particular conditions to be helpful for their
Excuse me, ma'am.
life cycle to continue.
Excuse me, ma'am.
Yes.
Hello, ma'am. Ma'am,
Yes.
what is meaning of corpse? Corpse.
Dead body.
Dead body. Okay, ma'am.
Dead body is called the corpse. Uh, So we are saying that the temperature plays an important role in developmental stages of these particular blowflies or the beetles. This particular thing will be mainly uh, located like geographical location. It is an indoor or outdoor, or it is exposure to sun or shade, or a time of day, and which season this body is lying in that particular place. These are all the conditions which make an important thing in the investigation. And heat generated by the maggot mass is also makes an very important role to identify the things. And whatever the tissue which is available in the decomposed body is the food source for them, like liver, heart, lungs. Majorly, they use this type of tissue to grow on that particular body to continue their life cycles. And they also release the various toxins near to that uh, dead body. And we also have an obstructions like plastic sheets, water, or other things may obstruct their particular life cycle to continue. But on the whole, it gives an lot of evidences to particular this thing to be uh, to collect the sample of that particular um, taphonomy to study of that decomposed body insects which have been visited already. And the collected insects also gives the most of the evidence. And other than the things whatever we need to calculate that particular body depth, we even follow the microbial forensics, forensic anthropology, forensic odontology, forensic biology, forensic DNA fingerprinting, and forensic serology. So these are the different branches of things, whatever the collected material have been got from that particular death of that body. We will give it to these many branches of serology, anthropology, forensics, and fingerprinting. And this way, we will, on the whole, have an very specializations required for this forensic science to be studied to get the evidences of that particular body. And we also have an forensic psychology also included, toxicology, ballistics, cyber and digital forensics, document fingerprinting, and forensic biology. These are the branches which makes an immediate study of this particular thing. How many? So in this particular study, we make an the PMI rate to be followed with the elapsed time from death to the discovery of the cadaver, and flies which are especially califorids and sarcophagids can be vital in determining the postmortem interval. And this premises of flies will begin oviposition as soon as they discover any body surrounding that particular environment where they are living. And the succession on a corpse is predictable, beginning on the particular oviposition as soon as they discover the body. And this will be helping in controlled rearing, and it also helpful for the collect the sample. So when we see this particular determining the postmortem interval, we say that this particular adulthood to confirm the species ID and also the insect opportunity to that particular area and the temperature occurring in that particular area and the evidence that might corroborate or the contradict to that place. So actually the stages of decomposition, usually we will calculate with the uh, four stages are being particularly discussed in this particular way of conclusion, that is fresh body and the bloated body, decayed body and the dry body. And the fresh body we will say that immediate body where they will find don't find any odor, and time after the death we see is of 1 to 3 days, and there we will see the eggs on the body. And bloating stage we'll see the swelling occurs on the body of the death, leaking fluids will be observed, and odor is also very much emitted from that body. And the calculated 2 to 7 days, and the larval stages we see inhabited on that particular body. And decayed body, when we see it emits a strong odor putrefaction that may be of 1 to 3 weeks, and all stages we will find in this on the body of this particular corpse. And this last stage is the dry, and where the bones we will find, and empty pupae and beetles are found, and it may be concluded that it may be lying there with the 1 to 3 months. So studying this entomology, the scope of entomology we can be of what we are saying is that best of a product dealer are the pest controller. Lot of pest controllers are being introduced into this.
Excuse me, ma'am. Hello. 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 Excuse me, ma'am.
Excuse me, sir. Sir, ma'am may be connecting to YouTube channel.
Yeah. Okay. I had doubt. Okay. I'll ask.
ಏನಾಯ್ತು ರಾಜೇಶ್, ಮುಗೀತಾ ಹೇಗೆ?
ಹಲೋ, ಐ ಥಿಂಕ್
ಏನಾಯ್ತು ಮುಗೀತಾ?
ಮುಗೀತ್ ಅನ್ಸುತ್ತೆ. ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ
ಹ್ಮ್
ಹೋಗಿ ಸೇಮ್ ಲಿಂಕ್ ಅಲ್ಲಿ ಸೇಮ್ ಪಾಸ್ವರ್ಡ್ ಅಲ್ಲಿ ಹೋಗಿ ಲಾಗಿನ್ ಆದ್ರೆ ಮುಗ್ದೋಗಿರೋ ಕ್ಲಾಸ್ ಕಾಣಿಸುತ್ತೆ ನೋಡಿ, ಕೇಳ್ಸುತ್ತೆ, ನೋಡ್ಕೊ.
ನೀನ್ ಲಾಗಿನ್ ಆಗಿಯಾ ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ? ಅದಕ್ಕೆ
ಆ,
ಲಾಗಿನ್ ಆಗೋದಿರುತ್ತೆ ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ.
ಪಕ್ಕಕ್ಕೊಂದ್ ಲಿಂಕ್ ಇದೆ ನೋಡು, ಅದಕ್ಕೆ ಕ್ಲಿಕ್ ಮಾಡಿದ್ರೆ
ಹ್ಮ್
ಸಬ್ಸ್ಕ್ರೈಬ್ ಕೇಳುತ್ತೆ. ಅದಾದ ಕೂಡಲೇ ಇವತ್ತಿನ ಕ್ಲಾಸ್ ಬರುತ್ತೆ.
ಓಕೆ ಓಕೆ.